Alrighty, hello everyone. Well, before we start and continue talking about membrane proteins in our last video, I had an equation up here that describes the chemical potential of a given substance moving across the membrane, where it's equal to the chemical potential. The chemical potential is equal to the chemical potential at standard state, plus the gas constant times the temperature times the Ln of the concentration of that substance. I had missed the temperature on that particular equation, right? Okay, so in this video, we're going to continue on talking about the transport of substances across the membrane, right? Passive diffusion, right? That does not require or is not coupled to an endergonic process and is really dependent on the chemical potential across the membrane, as well as the concentration of those ions and substances on either end of the membrane. So we'll discuss more about ion channels. On our last video, we ended talking about the KSCA potassium channel, right? And how it has the selectivity filter towards the exit of the channel that has the sequence TVGYG, right, which is a signature sequence typically found in potassium channels, and how it was selective towards potassium and not any other ion that may be of similar size, and that's because it coordinates or it binds to its carbonyl oxygen as it's moving through. Well, those ion channels are typically gated, right? So they open and close and respond to a stimuli, aside from differences in concentrations, they are gated. So they open in response to a stimuli. Some are mechanosensitive channels, and those open in response to local deformation in the lipid bilayer. An example of that is like touch or sound. Some are ligand-gated channels, and they open in response to extracellular chemical stimuli, such as neurotransmitter. Some are signal-gated channels, and those typically open in response to calcium or some other signaling molecule, right? Some are voltage-gated channels, and those typically open in response to changes in the membrane potential. Okay, so we will consider KV voltage-gated channels. Right? They have some resemblance to the KCS potassium channel that we talked about last time, but they typically have 4 different subunits, right, that are identical, but all subunits of those potassium voltage-gated channels contain around 220 residue N-terminal domain. So here's the N-terminal domain, which is about 220 residues. It also contains another, its C-terminal domain is about 150 residue. They're both in the cytoplasmic or on the inside of the membrane. And then it has 6 different alpha helices that are embedded within the membrane itself, right? Labeled S1 through S6, right? And on the bottom figure, it is showing you 2 of the 4 subunits, right? You have the N-terminal domain, and in KV channel, that N-terminal domain contains a conserved 100 amino acid residue sequence known as T1, right? That T1 is around 100 amino acid residue, and it's a conserved sequence in KV potassium channels. All right. And then it has 6 different alpha helices within the membrane. It also has, if you notice, between the S5 helix and the S6 helix, there's this loop known as the P loop. And that P loop is basically the selectivity filter that has the sequence or the signature sequence TVGYG found in potassium channels. All right. Well, it's a voltage-gated channel, so it's going to open in response to changes in the chemical potential. Okay. So what it has is one of its 6 helices, specifically the S4 helix, right? which is typically about 19 residues long. It's mainly composed of hydrophobic amino acid residues, but it has 5 arginine residues that are positively charged, of course. So here's 1, 2, 3, 4. It is showing us 4, all right? So it has arginine residues, and between each arginine, one arginine residue and the next, there's about 3 hydrophobic amino acid residues, right? So the S4 helix is otherwise hydrophobic, except for those 4 arginine residues. Okay. So when the membrane potential increases, meaning that within the inside of the cell it becomes more positive or less negative, that S4 helix is pulled outwards towards the extracellular side of the membrane. And as you may think, the S4 helix is connected to the S5 helix. So what it will splay that S5 helix as well, right? So here's just another example of that KV channel and how it is a tetramer composed of 4 different identical subunits, right? It's alpha helices span basically the lipid bilayer, and its T1 domain is within the cytoplasmic side. And all 4 different subunits form that inner channel for potassium to basically pass through. Okay, so when the membrane began to depolarize, meaning it's more positive towards the inside, that S4 helix, which acts as a voltage sensor and has the positively charged arginine residue, will end up being pulled towards the extracellular surface. And it will pull with it the S4 and S5 linker helix, right? But the S5 is also connected to the S6, which will splay it, and it will enlarge that inner cavity or channel to allow the entrance of potassium to pass through the channel, right? So it opens up and closes. And here's an example of it being open versus closed. And notice the S4 and 5 helix change. Okay, so as the cell, or as it begins to repolarize, when the inside of the cell is more negative, the S4 helix will basically drop back towards the inside, towards the transmembrane domain, towards the cytoplasmic side, and then it will press down and it will close the entrance for the potassium ions to pass through, right? So it opens and closes in response to changes in the overall potential being positive versus negative. Another example of selective proteins that are selective towards specific ions are known as aquaporins. And just like the name implies, they mediate the movement of aqua, or water, across biological membrane at a very fast rate, 3 × 10⁹ water molecule per second. There are about 13 known mammalian aquaporins. They're expressed in high levels in tissues that rapidly transport water, and such as kidney. In fact, the kidney itself has 7 different aquaporins, each with a specific function, all right? And they're very specific that they do not permit the passage of other solute or ions. Now, there are some that do allow other ions or solutes to pass through, such as urea, but for the majority, they only allow water to pass through. Plants have 50 different aquaporins, right? So they're very common in nature. So if we take a look, and they don't allow the passage of other solute, and we'll talk about one example that can actually differentiate between water and hydronium ion. And if you think about water versus hydronium ion, they differ by a proton, right? So that it's very selective. And one of the most characterized aquaporins is AQP1 aquaporin, right? It is a homotetrameric, so it has 4 different subunits, and they are colored within here, right? Each of its subunit has mainly 6 different alpha helices within the membrane, and then a shorter helix, right, that lies within the membrane. Okay, so part of the protein is exposed to the outer surface, part of the protein is exposed to the inside of the cell, but if you've noticed in all the examples we've covered so far, those transport proteins are mainly alpha helical or beta sheets, and that secondary structure of the protein, that part of the protein is embedded within the lipid bilayer, right? And we use the term transmembrane proteins for proteins that span the entire lipid bilayer, where a portion of the protein is to the outside, the majority of it is within the inside, and a portion of it is exposed to the outside. Well, if we take a look at AQP1 aquaporin, right? At the center, at the narrowest point of the protein, there's 2 specific highly conserved asparagine residues that will hydrogen bond to water, a single water molecule, right, via its amino group. And as the water molecules are passing through, there's also another histidine and an arginine residue that will hydrogen bond to water, as well as the backbone carbonyl oxygen. So if we take a look at the narrowest point of that protein, we'll see the 2 highly conserved asparagine residues that will hydrogen bond with a single water molecule as it passes through. So it's quite selective for only water molecules, but as the water molecules are passing through, there's also a histidine and an arginine residue nearby that will form hydrogen bond to that water molecule that is passing through. All right, so we talked about a number of different membrane proteins that act as channels for the passage of different substances across the membrane. Well, a different type is known as connexins, which form channels, but those channels act much like a gap junction between cells, right, that allow the transport of smaller molecules, whether they're ions, amino acids, sugars, or nucleotides, right? Some transport protein now undergo conformational changes, and we'll talk about those. So not all type of proteins form these channels within the membrane. Some undergo conformational changes, right? And one example is the erythrocyte glucose transport, known as Glute E1. It has 12 different membrane-spanning alpha helices, which are shown right here, right? And it has, and notice both its N and C terminal domain are within, towards the inside of the membrane, right? And it has a glucose binding site on either end, right? One glucose binding site exposed to the inside of the membrane and another glucose binding site exposed to the outside of the membrane. And John Berendt proposed that Glute E1 has 2 different conformations. What he did, he added a propyl group to carbon 1 of glucose, right? And notice that it will bind to the extracellular surface. And that's it. And then if he added a propyl group to carbon 6 of the glucose molecule, it will bind to the intracellular surface. So what he proposed, that the glucose has 2 different faces. On the extracellular cell surface, it requires the contact of the oxygen on carbon 1, and the one that faces the inside of the cell requires the contact of oxygen 6, or the oxygen off of carbon 6, right? So the mechanism, or how it's transporting glucose, occurs as follows. We have a glucose molecule that will bind to the Glute E1 protein at one side of the membrane, right, on the outside of the cell. it will then the protein, once it's bound, the protein undergoes conformational changes, which exposes the other side to the inside of the cell. And then the glucose molecule is transported, and then the protein goes back to the original conformation, right? So glucose binds on one face of the membrane, the outside, and then the protein Glute E1 undergoes a conformational change that opens the binding site towards the inside, thus releasing the glucose molecule, and then glucose dissociates, and then the protein goes back into its original conformation so it can accept another protein. So it can accept another glucose molecule. So here's just another example of it, all right? You have the Glute E1, binds glucose, glucose is bound, so it undergoes conformational changes that allows glucose to dissociate, and then it goes back to its original conformation. All right, so we're going to look at an example that will, okay. What is happening? It is not clicking. Okay, not sure why it's stuck. Okay, let's make it. Give me a second. Okay, so one example is the insulin receptor protein, right? So insulin binds to its receptor protein, which we're going to talk about at a later chapter, right? That triggers a signal that allows those vesicles that are found towards the inside of the membrane to basically fuse with the plasma membrane itself. And what that happens is increases the level or the amount of glucose transport proteins on the plasma membrane, right? And the number, that increase in the glucose transport protein on the plasma membrane is triggered by insulin binding to its receptor protein. All right. And then glucose transport protein will allow glucose molecules to pass through. Now, when the levels of insulin drop once again, those glucose transport proteins will, end up reforming those small vesicles by endocytosis, right? And then those smaller vesicles will basically fuse to form larger endosomes, and then they'll butt off and form these smaller vesicles ready to bind or to fuse once again with the membrane once the level of insulin is high. Okay, so transport proteins can be categorized also based on, do they allow one specific type of substance to pass through in one direction, or two different types of substances to pass through in the same direction or opposite direction, so as uniport, symport, or antiport, right? Just like the name implies, symport, two different substances are passing through that transport protein in the same direction. Uniport, it's a single substance that is passing through in one direction, and in antiport, there are two different substances or ions that are passing through in opposite directions simultaneously at the same time. So now, on our next video, we'll be talking about active transport.